Good morning, everyone. Could I have picked a longer talk title? Also, I didn't know that I was going to be the first, actual first speaker. So, usually you save the best for last. It's interesting. Good morning. So today we're going to talk about cloudifying pipelines and making them enterprise grade, make them able to run at a large scale, and how to do that with Kubernetes. So a couple caveats first. Everything in this talk is public, but the opinions are my own, not my clients or customers. The talk references a lot of Microsoft services, but don't get up and run away if you're an AWS shop, because everything that I talk about is very applicable in the other clouds as well. And this content spans a lot of experience from industry, academia, and beyond, so there's something here for everyone, I hope. Just quickly, a little bit about me. I am a computational biologist by training, but I do a lot of cloud architecture. I really specialize in building very large-scale infrastructures for pharma and biotech, and really specialize in organizing data and then overlaying compute and getting your stuff up and running. And the goal is making things run bigger, better, faster, stronger, and in a more automated way. In addition, I'm a faculty member at the University of North Carolina at Charlotte in the Cypher Center, where we do infectious disease research, and I'm the author of Genomics in the Azure cloud. If you see some Genomics in the Azure cloud pig books laying around, they are free. Feel free to grab them. They cannot go home with me. I'll put some more out in a little bit. And no, you do not get to pick the animal. So Geraldine got a lovely pufferfish, and I got a big brown pig. I'm a Microsoft certified trainer and a Microsoft MVP awardee for my efforts in cloud, so obviously I'm a bit Microsoft biased, but I'm happy to answer questions across clouds. Today we'll talk about cloud data organization, and then why that's important in getting your data organized before you do anything else. And then we'll talk about Kubernetes and what is that and why you might want to use it. And then we'll also talk about an update to Tuple's Kubernetes platform that's specific to bioinformatics called Ahab. So cloud data organization. Why would you want to use cloud? Obviously, lots of people in this room are already using AWS or Azure or Google Cloud Platform for various purposes. But maybe for those of you who are not, the reason that we want to use the cloud are for, one, elastic scalability. So we can spin up very large compute, meet the needs that we need right now, and then spin things down, hopefully saving cost. We can also elastically grow any sort of storage that we may have, right? We don't necessarily have to go and buy a bunch of equipment. The idea that you're going to run out of storage space is just really non-existent in the cloud environment. And using cloud tools, we're able to orchestrate very complex data analysis and transformation tasks and automate these repetitive tasks. And a real goal for me when I work with clients is a bioinformatician should not have to be clicking a run button all the time, right? You have better things to do with your time, and you should be innovating and doing really cool things rather than having to babysit jobs, right? With cloud services too, because we're all sharing the same sorts of data lakes or same compute services, this enables a lot of analytical collaboration, and that really helps to break down silos that you see when we work with on-premise architectures. And all of these things, I think, help to improve scientific reproducibility, right? We can go back and reproduce what we've done because we have our DevOps and we have our code set up in a way that we can go back and see what was run, who ran it, and how it worked. So this slide is showing the kind of insides of an example data lake. This is a slide from a client of mine in the biomanufacturing space. They manufacture stem cells. And one thing that I do a lot of times with clients is we start looking at what are the different types of analyses that you run. And so once we figure those different types up, then we start thinking about how you all think about how you organize your data. So for this particular client, they wanted to organize by individual runs through their manufacturing process, and then also by individual studies and projects above that. And then they also want to separate things in their data lake by clients, because they sometimes do things internally and sometimes do things for external clients as well. And so at your organization, it's not a one-size-fits-all. You should try and figure out exactly how do you all think about your data. And the reason you want to get your data organized is, one, so you can find your stuff, but two, if you start patterning out how you order and organize your data lake, then the compute services can also follow that pattern and go and find things. This helps enable lots of automation later on, which I hope you'll see. So why this matters, and in addition to compute services being able to find the data, this enables scalable queries across your data. So a lot of times we ask questions like, all right, for that patient that's in our clinical trial, what's the gene expression for them for this one particular gene of interest? But maybe we want to expand that, right? I've got 100 patients in a clinical trial. I've got 1,000 samples. I've got whatever, and I want to ask questions across that data. So how many of the samples in study B have gene ABCB1 expression over 10 transcripts per million across all the RNA-seq analyses, right? Today, if you don't have your data organized, you have to go find all the individual output files, right, and then go cobble them together, write a lovely Python or R script, and then hope it works, and then now you've done this large manual effort. But once we get cloud data organized in such a way, we can use other services such as Azure Synapse Analytics, which is a data warehousing platform, or Databricks to go and read directly from a data lake, and we never have to like put things in lovely tables. We can read directly from them. So I think that's very powerful to really being able to ask very large questions across your data. So once you have your data sort of organized in a data lake, now how do we do stuff with it? And there are lots of compute services, but today we'll talk about Kubernetes. For those of you who are not familiar, Kubernetes is a container orchestration platform. So if you're used to using things like Docker, you can take Docker containers and orchestrate how they spin up and spin down in a Kubernetes environment. And there's lots of things that you can control in that environment that makes things work really well in bioinformatics. Namely, we can specify different resource needs by node pool. I'll talk about what a node pool is in just a second. But for different types of tasks, perhaps we need more RAM in one context, we need a lot of CPU cores in another context, and then maybe we need some GPUs in a different context. And we can do that and control those things individually in Kubernetes. So a very basic Kubernetes architecture looks like this. We have a cluster, and then in that cluster we have a what's called a control plane. The control plane manages the input and output of the cluster, and it tells the other nodes what to do, or tells the cluster what to do. We can spin up multiple different node pools. And usually I spin up a node pool per type of analysis. So perhaps we might have our RNA-seq node pool, we might have an exome pipeline node pool, and then maybe we have an AlphaFold node pool that needs some GPU nodes. And we can do this individually. We can also specify how many of each type of node that we want. We could let it be unlimited if you have unlimited money, but we also might want to limit that to, say, 20 RNA-seq nodes to keep the cost down, but while being able to process 20 things at once, right? Per node pool, we can also set different affinities, which are called tolerations and taints, and those enable us to say what types of jobs can be run on those types of nodes. So this is really useful if you share a Kubernetes cluster across different teams that have very different needs in the environment. With the cluster itself, you also get the ability to mount volumes. So volumes are data sources, and this is really, really, really cool with data lakes because we can mount an entire data lake as if it's part of the file system. So some of the other talks that you've heard where people were looking at, like Paolo's talk yesterday, talking about moving data around or using copying data with different file system fuse tools. We don't actually have to do that with Kubernetes. We can mount the entire data lake directly and just read from it as part of our pipeline like it's part of the file system, like it's sitting on the virtual machine, right? We also have the ability to manage secrets and networking and config maps, and all of this kind of really works to make the system work in an automated way. So you can run Kubernetes on your laptop, right? And in fact, if you have a newer version of Docker Desktop, you can run a single node Kubernetes cluster on that laptop. But why would you want to use cloud Kubernetes, right? Not everything in bioinformatics is a pipeline. I know we're at a Nextflow conference, but sometimes stuff is like a duct taped together shell script that's calling some Python scripts, and you can't run it on a Tuesday if it's raining, right? And so we want to make these sorts of pipelines a bit more robust and reliable, and Kubernetes can do that because it runs the same thing over and over and over again. So it adds a bit of those capabilities. Not every pipeline has the same needs as I was just saying, and because of this, we can take advantage of the supposed limitless compute capacity that we have in the cloud providers. We can also take advantage of cloud networking, which is blazingly fast, and then also the security that automatically comes out of the box with cloud providers. One thing that's really cool, I think, about Kubernetes is because it's an open source platform, the cloud providers have done a really good job of making Kubernetes just work without a lot of proprietary, secret sauce. So in Azure, if you use Azure Kubernetes service, that's cool. Any of the YAML files that you make for that, you can also apply them over in an AKS or EKS instance in AWS, and they should just work, except for some slight things around like VM or SKU naming for the VM types, for example. So it's really cool that it makes things very portable as compared to some of like batch services, for example, that operate in the same way, but they're not compatible between the cloud providers. So if you're in a multi-cloud environment, Kubernetes kind of works a bit better. And then lastly, like I mentioned, we can mount our data lake as a volume and operate as if it's part of the file system, which makes things a bit easier so that we don't have to worry about copying data around and writing the logic to do that. All right, and the last part of the talk, I'll mention Ahab. So for those of you that are not familiar with Ahab, it is our Kubernetes platform that allows for bioinformatics, for you to scale bioinformatics workloads. And we're now releasing version 2.0, which simplifies a lot of the infrastructure, and it makes it much easier to use. So Ahab is a tool where the goal is that you just BYOD, and that's Docker container, not drink. We bring your own Docker container. You make very minor changes to it, which I'll talk about in just a second. And it works with a variety of pipeline tools. Obviously, this is a Nextflow conference, so you can use Nextflow, but you could also use SnakeMake or even just a shell script or other tasks as well. This fully integrates with data lakes, as I mentioned, and it comes with an easy-to-use API that allows for you to submit jobs through a task scheduler and then update those jobs as well. So how does it work? When we build a pipeline, you bring all of you professional pipe layers, we bring our pipeline and grab your code, your pipeline scripts. We're going to bookend that code with a few commands from the Ahab CLI or Python package. The first is we got to get some job information, and the last is we need to update the job information if it completes successfully or if it fails. And then we modify the code just slightly so that it can accept the inputs from the API. And then we want to build a Docker image around that, and then publish or push that Docker image up to either Azure Container Registry, if it's a private image, or Docker Hub or other container registries. So in the example RNA-seq pipeline that everyone's been showing, I know it's a little hard to see. The only thing that we're changing is 4 lines. The 4 input parameters, where normally we're handling base directory and whatever, we just replace those with the environment variables from the API response, and that tells us exactly where in the data lake to go get the input files and the reference information, and also where to write the outputs back. We also have one modification is to use a run shell script that calls the Ahab API, running. Then the next thing is it runs the Nextflow pipeline, and then depending on if the Nextflow pipeline fails or succeeds, then it updates the job accordingly, stamping it as completed or stamping it as failed, and then captures some information about the outputs. So how does it work? The Ahab system includes an API. The API every minute will go and ask the database if there are any pending jobs per job type. So in this situation, there are 42 RNA-seq jobs. The API will communicate with the control plane of the Kubernetes environment, spinning up the appropriate number of nodes to handle that task. Once those pods spin up in the nodes, the containers it will grab the appropriate container for that. And then the containers will execute the command that we need. So for example, it'll kick off by running that shell script that I showed on the last slide. And then the Kubernetes environment can then, or the pod itself can actually communicate directly with the data lake as if it's part of the file system, grabbing the reference data and the input FASTQ files, for example, and then writing the results back directly. And it's very, very fast. And this is kind of what a job object looks like in the database. We have an ID for the job. We have it specifies what the job type is, and then we have a status history of when it was created, when it's pending, when it was started, and if it completed or failed. And then also you can see that we have some input information about the transcriptome and input reads, and then also the output directory as well. And then notice we've also stamped this with the specific image that was run. Again, hopefully with the goal of scientific reproducibility, we can go back and see, all right, this was run on this date, here's how long it took to run, and here's the very specific image, including the tag that it was run on. And once we start spinning up things in Kubernetes, like I mentioned, it auto scales. So when Kubernetes is sitting there by with no jobs, it's just a tiny little virtual machine that's continually running to run the control plane, very cheap to run. And then whenever we have, let's say, 42 RNA-seq jobs, it will then balloon out, spinning up the appropriate number of nodes in the node pool. They'll each kick off individually and then either complete or fail individually, and then they'll spin back down once the tasks are done. And all you pay for is the amount of time that you used. So some final thoughts are the cloud helps us scale through data lakes. Once you have your data organized, then we can do stuff with it. And once we can do stuff with it, that includes things like very scalable cloud compute services such as Kubernetes that I've discussed today. And Kubernetes works really well in the bioinformatics world because computational tasks take a long time to run. They're oftentimes it behooves you to put them in a Docker container because you got weird versions of everything. And it really helps to then scale those pipelines and not have a bioinformatician having to click a run button every single time that there's new data that comes in. The idea is that it could be fully automated. And so coming soon to Ahab, we're going to include some additional pipeline examples in the GitHub repository, which is all open source. Add in some additional pipeline CLI updates and a Power BI dashboard template so you can visually see what your task statuses are. We're continuing to add additional support for GPUs, different types of GPUs in the Azure stack, and then also integrate with other Azure compute services as well. And with that, I'm happy to take some questions, and here's my contact information. Feel free to find me on LinkedIn.
Thanks, Colby. We have some questions here. So what storage file system do you use for the pipeline work directory?
Yeah, so it's Azure Blob Storage, but specifically Azure Blob Storage with the hierarchical namespace turned on, which turns it into a data lake. So it's the data lake service. It's the equivalent to S3, but it's slightly different underlying technology in the Azure stack.
Is this using the local Nextflow executor or Kubernetes executor?
This is using the Kubernetes, it's using the Nextflow executor inside the container, but it's a Kubernetes executor to run the control plane. So it's like it's being executed at 2 levels, right? The containers being spun up through the Kubernetes control plane, and then once the container spins up, then it's just running your normal Nextflow executor.
And a final question, is there an advantage in making different Kubernetes node pools per analysis type versus a single large node pool or node pool segregated on some other attribute?
Yeah, good question. One of the things that is beneficial in Kubernetes is because you can specify resource limitations per node pool type, it might be useful for, like, let's say an AlphaFold node pool to have access to GPUs, but not a whole lot of CPUs because it may not use those. But then maybe an RNA-seq job needs a lot of RAM because you're running very large genome or transcriptomes through that. And so yeah, it is better to put them in separate node pools because of the different resource needs of the different analysis types.
Thank you very much.
Thank you.